All right. Hello, everyone, and welcome to the moderated open discussion on the role of FUS in therapeutic delivery. I want to thank Dr. Wang for a really fascinating presentation. I think it sets us up very nicely for our discussion here. Just as a reminder for anyone who would like to participate, this is open to everyone here on the call, and I'll just ask that you please use the raise hand feature and wait to be called on to keep the discussion somewhat organized. So the goal here is really to shift gears a little bit and now move from what's been, I think, a very rich discussion about the role of FUS in immune modulation, and instead think about the more traditional way that we've tended to think about FUS, which is as a vehicle for enabling therapeutic delivery. And so I want to open up the floor to maybe answer a combination of two of our listed guided questions here. One being, what are the outstanding challenges, I would say broadly, with immunotherapies, but particularly novel immunotherapies like CAR-T that FUS can help to address? And also with that in mind, what immunotherapies have emerged as the strongest candidates for delivery with FUS? So maybe we can stage the discussion by starting with those two questions. And I'll open up the floor perhaps to Dr. Wang first, since his presentation just wrapped up.
I just want to thank you, Natasha, for organizing this session. It's really a pleasure and honor to present our work here. I will be happy to answer questions about our research and, of course, participate in this panel discussion.
Thank you so much. So, we just finished up talking about a very unique way that you all are using CAR-T cells. And so I want, going forward, I think, to really be able to focus this discussion both on CAR-T cells as well as the other sort of leading candidates in therapeutic delivery right now, which are monoclonal antibodies. There are a lot of groups here who have worked on some form of delivering checkpoint inhibitors. And so I think that this will sort of set us up to move forward in the discussion. And with that, I see Tanya Kuklova's hand up. So Dr. Kuklova, go ahead.
Thank you. So I have a question for Dr. Wang about homing of CAR-Ts. So I've heard that in lymphomas and leukemias, CAR-Ts have been extremely successful to date. However, not so much in the solid tumors. And one of the issues is homing. So they just don't home to the tumor. And so in your opinion, when you apply this hyperthermia and see enhanced activation and penetration of the CAR-Ts, is it that you're just turning on that gene and they do what they need to do, or do they also home better to that area if you do that?
Tatiana, thank you very much. That's an outstanding question. We actually, this particular paper, we do intratumor injection, try to, basically enhance the efficacy and efficiency of the, number of the cells at the local site. We are, of course, right now doing the intravenous injection. That's an ongoing project. Currently, we are just trying to preheat the cells so that they will already express the CAR on the surface, and we inject intravenously. And hopefully, within this window of the CAR receptor lifetime, they will be able to reach the site, and then we can turn them on again, when they reach the destiny. So that's the current, ongoing project. Definitely, that's a major issue. Probably Natasha will have a lot of discussions around this topic as well, how to deliver those CAR-T with high efficiency. But that's an outstanding question.
Excellent. Dr. Frank.
Yeah, I'd like to follow up on that as well. I mean, it is, at least from the CAR-T cells and at least the studies being done at NIH, they're all an intravenous injection. And because we're giving large numbers of cells in order to get any sort of delivery to a tumor. I'm not clear to me that, in all the cases, direct injection of a tumor may be fine for certain things and for proof of principle, but essentially you're going to have to come up with a way to essentially figure out a way to deliver this with systemically, because especially in metastatic disease, you're just going to have to deliver a lot of cells to a lot of places, billions and billions of cells. I mean, One issue is if you're going to heat it outside the body, why not try ultrasound over a vein and just essentially try to heat it on the way in to the body? And that may be another way to do it. The other question is, you could it pre-sonicate? I mean, as far as Tatiana's question is, you pre-sonicate, you change the tumor microenvironment in a way that there would be increased homing as well. But so one question, so the PNAS paper you had originally was with a mechanostretch receptor in the cells. Why not go back in that direction again with a calcium, change in calcium flux and everything else, versus going to a heat shock protein promoter? So that's really sort of, there's two ways to look at that. You don't have to worry about the heat. You can just pulse essentially and not increase the temperature in the tumor and still get an effect if you can turn on those CAR genes with basically with mechanical stretch changes in the lymphocytes.
Another outstanding question. Thank you very much. So of course, let me separate these two, into two parts. So that because you asked first about heat-related one, right? Which we are indeed doing that, preheat, and also we can use like some kind of water bath so that mice can take a, like a hot shower, periodically, so that we can enhance the homing, and then we can activate them. That's actually an ongoing project, which is like as Tatiana asked. And we are also, trying to enhance the like the cell capability or the proliferation, like by manipulating using CRISPR to knock out a metabolic pathway, so that they can become more robust. Actually, there are some already publications, coming out showing with manipulation metabolism, you can indeed enhance when the cell arrive to the target, they can, proliferate much better. So that's the heat part. Of course, I was trained as a mechanical engineer in the beginning. So mechanical stimulation is always my, like personal favorite, right? Our PS paper in 2018, we still use a macro bubble to amplify the signal, to turn on the piezo channel and then get some signaling. And we found out that's quite efficient, but if we take out macro bubble, which probably, will be needed for in vivo study, then the efficiency is just much reduced, and we couldn't, have the robust activation of the capsid. You can occasionally, but not that robust. So we are indeed trying to, engineer a different way, still using mechanical, manipulation, but the capsid seem to be not that strong and relatively noisy. So we are trying to, integrating with like end gate or synthetic biology to amplify signal at the same time reduce the basal background. That's another ongoing project. It has been, quite years. Seem to be a small, enhancement, but a lot of work needed to really reach a robust efficiency.
Thank you. And Dr. Price.
Thanks, Natasha. So I missed the first hour. So if what I'm about to say has already been covered, just go ahead and mute me, please. I'd like to make a pitch for the endothelium here, and I think it's related to the comments that have been going on with the CAR T cells. At least yesterday, I didn't see a lot of discussion about how focused ultrasound can change the endothelium with regards to its ability to recruit immune cells. We talk a lot about tumor cells, we talk a lot about the immune cells themselves, but of course, the endothelial cells are the gatekeepers. They're the ones that are essentially the cells that are responsible for trafficking immune cells from the circulation into the tumors. And, we also know that in many tumors, the endothelium is quite dysfunctional. It lacks chemokines, it lacks the correct homing receptors to facilitate immune cell recruitment. So we can set up a situation where we have a wonderful population of circulating effector T cells, but if the endothelium is not ready to receive those cells, we're not going to complete the cancer immunity cycle and get those cells in there. So it's related to this because I think there's an opportunity here in these situations with, adoptive T cell transfer and CAR T to use focused ultrasound, first of all, as a tool to modulate the endothelium and make it more receptive to recruitment of these cells. And, the word I think that's out there a lot in the community now is endothelial energy, similar to T cell energy. So, can we use focused ultrasound to reprogram the endothelium from an anergic state into an immunogenic state to now go ahead and better facilitate these types of responses. So anyways, I'll put that out there because I don't think it's something we talk a lot about in this community, you know, how FUS impacts endothelial cell function with regards to immune cell recruiting, how we can use it to drive that. And you know, the other thing to think about might be incorporating things like anti-angiogenesis therapies into this, because you know, therapies like VEGF trap, we know that they'll do things like normalize the vasculature. They'll make the microcirculation of a chaotic microcirculation in a tumor look more like a normal circulation, microcirculation. Well, the other thing that they do is they actually tend to limit endothelial energy. They can make the endothelial cells more functional with regards to their adhesion molecule expression. So, you know, maybe from the foundation standpoint, thinking of projects that integrate anti-angiogenesis therapies into them to, again, better facilitate immune cell recruitment could be something to think about as a community going forward.
Rich, I think you raised some really, salient points here. And just to comment on, whether or not this idea of homing and impact on vasculature was discussed, I'll just offer that it was touched on very briefly this morning, but I think that the overarching consensus was that there's still a lot we don't know, and there isn't necessarily definitive evidence of how we can modulate the vasculature, which is, I completely agree, a very important point, particularly as we move into the realm of trying to potentiate things like CAR T-cell therapy. So I see Dr. Wang's hand up. Perhaps he has a comment on this to further the discussion.
Yeah, I just want to follow up. I think that's a great idea, follow up on Richard and echo his comments, to manipulate and somewhat reprogram the vascular network, particularly, at the tumor site. That would be really wonderful. Although I still think perhaps relatively non-specific perturbation using ultrasound may not be that efficient and specific. So somehow if we can deliver genes through virus or what other means to genetically manipulate the vascular network, perhaps will be more specific and efficient. That's my personal opinion.
That's great. And in fact, I was about to open the floor for Dr. Frank's comments as well, since his group has done a significant amount of work in this space. So, right. Dr. Frank.
So the question, Rich, are you thinking about more the increased expression of integrins and changes in the surface expression of cell adhesion molecules and everything else, which are essentially for trafficking of these kind of cells, any type of cells almost? Even if we go to a sort of a stem cell approach, they're important, they're essentially to get the homing done, on top of essentially release of certain factors from the basically the cell itself, the endothelial cell itself. Because right now we've always looked at it as a sort of a totality, from a macroscopic view, and sort of tweezing out what the endothelial cell actually secretes, or actually is altered, basically what changes in the endothelial cell that results in it, say, for instance, producing MCP1 or VEGF or EPO, is really something we don't know exactly, other than we have ideas that it's driven somewhat by calcium and stretch receptors in the endothelium, but the same thing, the cell adhesion molecules come up very quickly and are necessary. And one of the things that I think we have to talk about or think about with sort of ultrasound techniques is how long can we maintain that elevation? Are they going to disappear in 24 hours, or is there something that we can do over multiple days? And I think that's really important. But is that what you were trying to get? I'm sort of directing this to Rich, but at the same time, that is essentially what I was thinking, that there is a period of time where the surface antigens really get expressed and really get changed on the endothelial cell. And we can see that by histology. The real question is, can we accentuate that? Because that's going to be essential for treatment of autoimmune diseases. I have a background in autoimmune diseases, and this is basically how cells and T cells get into the brain and different parts of the body. And this is essentially what's going to have to happen with CAR T cells and treatment, or genetically engineered stem cells and treatment of tumors.
Yeah, no, I agree entirely, Joe. I think, another way to maybe interpret my comment is in those situations where we have a weakness of response, maybe we should be looking to the endothelium a little bit more. And that very much could be related to your comment about the transient nature of the expression of the molecules. They need to be on at the right time to get the cells there. And if they're not on, you got to figure out a way to put them on. That's pretty much it. Yeah.
Quick follow-up. Maybe we can, deliver like genetic material to these, endothelial cells so that we can control relatively, lasting expression of ECAM, ICAM, those adhesion molecules, the CAR T.
Dr. Wang, your audio was fading in and out just a little bit. So we lost you at delivery of genetic material.
Sorry, is it better now?
It's a little bit better, yes.
Okay, so basically what I was saying is whether we can, deliver genetic materials to these endothelial cells, and we can control the relatively long-lasting expression, for example, ICAM, ECAM, those adhesion molecules, so that they can recruit the perfused, CAR T to the local sites.
It's an excellent idea, and I know that Dr. Price's lab has done some work in this space and is interested in that as well. So Dr. Arvanitis, I see your hand has been up.
Hey, hi everybody. I think that using the genetic toolbox is always like a tempting option, and is always will produce more, to some extent, durable results. But I do think that the evidence that has been collected, especially in the brain by this society, clearly suggests that ultrasound mechanical effects can mediate significant responses from the endothelial phenotype. And there are several studies, and I think maybe that will be of interest to the foundation, that we have seen, and including Richard, and you have shown that the timing that we deliver drugs, either those could be like antibodies or could be CAR T cells, as has been shown a few years ago by a group by Kolervahin's group. It's extremely critical and produces hugely different responses. This suggests that ultrasound alone might be sufficient to promote robust responses and facilitate therapeutic trafficking, whatever therapeutic you want to use, and also facilitate homing. So I think using the genetic toolbox is great, because you can promote the changes that you want to promote. But at the same time, ultrasound and microbubbles or heat can also induce significant responses that can facilitate trafficking, which is a big challenge, for example, in the brain. And I think another suggestion, because I know the foundation is acutely interested in promoting interactions between basic scientists and clinicians, then that could be like a very good point moving forward for treatments that are already in phase 0 or phase 1 clinical trial. It might be a great opportunity there, in addition to try to identify the immune landscape. Maybe it's great to try to understand what ultrasound does to the endothelial phenotype. And there you just need to wait few maybe couple of hours, so you have transcriptomic level changes, and then that could provide a huge information, not only for the immunotherapy community, but also for the nanomedicine community that they're trying to make targeted molecules and therapy. So I think it will also be an impactful area of research to try to really understand what ultrasound, different modalities, exactly do to the endothelial phenotype. We know a lot about permeability, and that's most of the interventions that we do with ultrasound. They end up being named by changes in permeability just because this is what we observe first. But I think there is a wealth of things that are also going on, and many people in this call have contributed to that maybe moving to the clinical level, maybe this can be like a really nice field of interaction between basic scientists and clinicians to, for example, define time points that if we harvest the tissue, is very likely to observe any responses, if there are any, in the clinical setting.
I think that's an excellent suggestion, and I don't know if we have any of our clinical folks on the call here, but it would be really valuable to hear input about how that interfacing can occur between the preclinical and clinical efforts. And just to clarify, Dr. Arvanitis, I was curious, I think you make a very interesting comment about taking sort of a systematic approach to looking at the impact of different focused ultrasound modalities on the endothelial phenotype. I was curious about whether you're suggesting something akin to like the GBM consortium where several groups came together to assess something like this. Would that be a potential model?
That's something that we are really interested in. We have recently shown, and we are thankful to Foundation that help us move in that direction. We apply very safely hyperthermia in the brain. We've done a number, and that's like with closed loop control and image-guided therapy. We have built our closed loop controllers with microbubbles. So that's extremely important to have closed conditions, a closed loop control method, so you can really tune the mechanical and thermal stimuli. But then, going and supporting and going in depth, the Foundation provided the funds a few years ago and was very helpful, I think, to the community to understand immune cell trafficking. So maybe is another idea is to go and try different modalities and see at what exactly changes happen in the brain tumor microenvironment, for example, with respect to when we apply different ultrasound modes of operation.
Excellent.
And this can be, I think, one of the great comments. I think one of the good approaches this can happen is maybe we do spatial analysis transcriptomics instead of like bulk measurements, so we can really decipher the level and the location of things happen, because we might see a significant increase of molecules that this might happen in any case in the inflamed brain. So you don't really know whether they come from the endothelium or they come from the tumor. So I think really going and being specific about the specific cell populations that respond, I think it could be quite central here.
These are great suggestions, and I know that the people on the Foundation side are definitely taking notes so that we can circle back to some of this stuff during our future road mapping discussions. Dr. Koklova, I'm sorry to have kept you waiting, but I'd definitely like to invite you to share your question or comment.
Yeah, so it's sort of a question broadly to Dr. Price and broadly to everyone interested in endothelial cells. So discussion so far was focusing on cells, right, cars and things, but is there, what is, in your opinion, what changes in endothelial cells specifically, is it inflammation similar to enhancing homing of cells or something else that could enhance trafficking of molecules like PD-1, like antibody treatments or chemo even?
And I just want, before we open up the floor for that question, to mention that we have about 5 or 6 minutes left in this session and one more hand raised. So I just want our speakers to be mindful of the time if possible. Thank you.
You want me to try to answer that, Natasha, or do you want to take your next question?
Yeah. No, go ahead. Yeah.
Yeah. So Tanya, I was specifically thinking in the context of immune cell recruitment, not so much in terms of enhancing the trafficking of antibodies or other therapeutic molecules across the barrier. Yeah, and I think it's one of those things that's going to be dependent upon the tumor that you're looking at and where it's located. Some tumors don't necessarily have an anergic endothelium, but plenty of them do. And in those cases where we do have that, I'm offering that focused ultrasound actually could be a nice tool to, as has been discussed, genetically modify, for example, those endothelial cells to reprogram them into a more functional type of phenotype. Although Costas's points are well taken as well, I think near term, other approaches to modulate or to think more pointedly about how you modulate the endothelium with simpler approaches makes sense, and also is something that we can think about in the context of clinical trials.
Excellent. And Dr. Wang, I see your hand up.
Yeah, so I just want to make a comment on use the nanotechnology. Maybe like a polymer, like a Dangerma we use in our group, can make the targeting to the endothelium cell or tumor marker, plus the immunotherapy can bring this efficacy go to the local, like a targeting modality. So we have paper on the journal nanotechnology, use the E077E2 mouse tumor model to deliver antibody to the HER2 side. So if you're interested, you can join this like nanotechnology with the gene therapy, then use the tumor model to monitor this change. So this can be preclinical or more basically like a time point study to get the detail for the response cellularly or the humoral response. Use this technology plus the model system we already established.
Excellent. And you know, I think that's a perfect segue for these last few minutes of the discussion, as we've now talked about really the role of the underlying biology in terms of how we're going to be able to drive therapeutic delivery. But I wonder if any of our speakers have comments on of the delivery platforms, for instance, different antibodies that we've seen delivered in the literature. Do we see any candidates that are rapidly rising to the top in terms of being primed for clinical translation? And, you know, of course, the classes of therapy that I'm thinking about here are mainly checkpoint inhibitors. We've seen everything from anti-CTLA-4 to anti-PD-1, anti-CD47. The list goes on. But I'm wondering if anyone would like to comment on their experiences with these delivery platforms and what they think we have that's ready for translation and what might be missing in that realm. Just to touch on the topic of antibodies very briefly. Kostas, go ahead.
I think, you know, Richard, to go ahead with the clinical trials with the work that you've done with the antibody. I think that's I think the best and most rationally defined study. I think you should go forward with that, with targeting macrophages in the GBM space. I know that you cannot say that, so I'm saying that.
Oh, you're talking about our work.
Yes.
Well, that's very kind of you. I appreciate that. But you know, and you know, I guess that sort of just speaks to the fact that you know, depending on study design, something we're going to get to as well is what we need in the way of endpoints and monitoring methods as well. And for instance, that study employed immuno-PET monitoring to really be able to show that we were delivering antibody to the target site. And in my experience, this is a question that clinicians often ask with the mechanism like BBB opening. And so hopefully in future discussions here at this workshop, we can also touch on, for really prime opportunities like antibody delivery, what more we need in the way of tools to help frame some of those questions. Any closing comments? We have about a minute left. Final questions or comments? Okay, well, I would like to thank again our presenter, Dr. Wang, as well as all of the participants here in this discussion for being so engaged. I wish we had significantly more than 30 minutes of time to touch on the wide variety of therapeutic options that we could have discussed here, but thank you all so much for your participation.